Replication. DNA replication is a process by which DNA makes a copy of itself during cell division. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. Second, this is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding a complementary bases of DNA together. Third, the separation of two single strands of DNA creates a Y-shape called a replication fork. The two separated strands will act as templates for making the new strands of DNA. One of the strands is oriented into 3 to 5 direction. This is the leading strand. The other strand is oriented in the 5 to 3 direction. This is the lagging strand. As a result, to their different orientations, the two strands are replicated differently. A short piece of RNA called a primer comes along and binds to the end of the leading strand. The primer acts as the starting point of DNA synthesis. Sixth, DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases to the strand of DNA in the 5 to 3 direction. This sort of replication is called continuous. Lagging strand. Fifth, numerous RNA primers are made by the primase enzyme and bind at the various points along the lagging strand. Sixth, chunks of DNA called Okazaki fragments are then added to the lagging strand also in the 5 to 3 direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. Once all the bases are matched up, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. Ninth, the new strand is proofread to make sure there are no mistakes in the new DNA sequence. 10th, finally, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands. 11th, the result of the DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule, half is brand new. 12th, following replication, the new DNA automatically winds up into a double helix.
Transcription. To begin transcribing a gene, RNA polymerase binds to the DNA of a gene at a region called promoter. Basically, the promoter tells the polymerase where to sit down in the DNA and begin transcribing. Each gene has its own promoter. A promoter contains DNA sequences that let RNA polymerase or its helper protein attach to the DNA. Once the description bubble has formed, the polymerase can start transcribing. DNA double helix must unwind near the gene that is getting transcribed. The region of opened up DNA is called transcription bubble. Transcription uses one of the two exposed DNA strands as a template. This strand is called the template strand. The RNA capping and splicing. Number 7. The RNA may remain in the nucleus or exit to the cytoplasm through the nuclear pore complex. The RNA produced is complementary to the template strand and is almost identical to the other strand, called the non-template or coding strand. However, there is one important difference in the newly made RNA. All of the T nucleotides are replaced with U nucleotides. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA nucleotide. Polymerase adds RNA nucleotides, which are complementary to the nucleotides of one DNA strand.
Translation. Messenger RNA translated into protein. Translation is the process that takes the information passed from DNA as messenger RNA and turns it into a series of amino acids bound together with peptide bonds. It is essentially a translation from one code, nucleotide sequence, to another code, the amino acid sequence. The ribosome is the site of this action, just as RNA polymerase was the site of the mRNA synthesis. The ribosome matches the base sequence on the mRNA sets of 3 bases, called codons, to tRNA molecules that have the 3 complementary bases in their anticodon regions. Again, the base pairing rule is important in this recognition. A binds to U, while C binds to G. The ribosome moves along the mRNA, matching 3 base pairs at a time, and adding the amino acids to a polypeptide chain. When the ribosome reaches one of the stop codes, the ribosome releases both the polypeptide and the mRNA. This polypeptide will twist into its native conformation and begin to act as a protein in the cell's metabolism. The steps in translation are: the ribosome binds to mRNA at a specific area. 2. The ribosome starts matching tRNA anticodon sequences to the mRNA codon sequence. Number 3. Each time a new tRNA comes into the ribosome, the amino acid that it was carrying gets added to the elongation polypeptide chain. Number 4. The ribosome continues until it hits a stop sequence. Then it releases the polypeptide and the mRNA. Number 5. The polypeptide forms into its native shape and starts acting as a functional protein in the cell.